fun,
but
yeah.
like a mini image along with the chat box. We can't define, we still have to speak from the like ethos, but
Yes.
if we could have her in her there, so that if she unusually wants to talk, like here or
I'm not going to promise you the chat box because I'm not sure how they did it yesterday, but let me see if they can do it.
Oh, the chat box, they did it yesterday.
Yeah, I know they did.
Yeah.
I got to go see if they've got it set
What's
up.
more important is the chat box. That's
So if I can do that side by side, I will.
Yeah, it's not just the chat box. Or the chat and the Q&A.
Okay.
Either
But I'll just have her program audio up so she can talk.
Yeah, that's fine. Thank you.
That's our fallback, but I'll see if I can get it for you.
Okay, that's fine. All good.
You're going to hang on to that for Zoom.
Yeah, I will, but if you need it, you can steal it. Yeah.
Well, let me know when you want it.
Oh, right. There's that. Ugh. Thank you.
Yeah, beautiful car.
Okay. Start talking. I know I'll sit down.
Start talking.
Yeah.
All right. Looks like we're all back. So I'm Kayvon Pedram. I'm a group leader here. I'm really pleased to introduce Celeste Nelson, who's a professor at Princeton. She'll be joining us remotely and telling us about the evolutionary differences in organogenesis. Celeste, take it away.
Thank you. Can you hear me okay?
Yes.
Okay, awesome. Well, last week I thought I was being clever by getting my bivalent COVID booster a week before needing to travel. I thought that meant that any side effects would be over by now. What I didn't anticipate is that I would get very dizzy from the booster shot, faint, and land really, really hard. So my ribs are fractured or badly bruised in 2 places, and I badly bruised my lower vertebrae. So I apologize for not being there in person. I can't travel right now. I'm being propped up by pillows, but I'm really grateful to Ron and Jennifer for the opportunity to participate, and to Janine and John and the rest of the staff for coming up with the last minute accommodations. So today what I thought I'd do is to share some of my group's work that sort of fits within these concepts of 4DCP, specifically trying to understand the mechanical processes that fold epithelial tubes during our favorite model system development, the lung, with the ultimate goal of using the processes that we uncover to build tissues ourselves. So the lung is my favorite organ. And it's my favorite organ in part because its function is intimately coupled to its form. And you can remind yourselves of those functions of the lung every time you take a deep breath, and how closely those functions are tied to the anatomy of the organ, right? So to breathe in, you contract your diaphragm, which increases the volume of your chest cavity, which decreases the pressure and allows air to flow through epithelial-lined tubes of airways until it reaches the alveoli, which are these cup-shaped, thin-walled epithelial surfaces that allow gases to diffuse between the air spaces and the lung capillaries. So we've been really interested in understanding how these different tissues build themselves with the purposes of actuating those developmental mechanisms ex vivo, right? So how are they built? Well, one classical approach to investigate this question of how these tissues are built is to identify the molecules that are involved in morphogenesis of them, which is an approach that has been pioneered by luminaries in the field with a specific focus on identifying the signaling pathways that are essential for building the airways and the alveoli in the mouse lung as a model for human lung development. And dozens of molecules, of course, have been identified. But similar to the example that Hernan gave yesterday, this diagram here is akin to a parts list. It tells you the genes that are required, but if I remove the cartoon behind all of these genes, you wouldn't know which tissue is being built. Right, so this parts list is not enough to tell you how the epithelium is sculpted into the final structure of the lung. Right, so I'm a bioengineer by training, and as a bioengineer, I'm most interested in understanding how tissues are built physically. So my group has taken an orthogonal approach and focused really on the biophysics of the process. Right, so if we think of an epithelium as either a flat sheet or a hollow tube, then it's basically a material, and that material has some mechanical properties. It'll either be elastic or viscoelastic, and it'll respond to forces, either those that are generated internally or those that are imposed upon it, to fold itself into the more complex structures that we see in organs like the lungs. And as Xiaohe mentioned yesterday, in contrast to the salivary gland, the epithelium of the embryonic lung is a single sheet of epithelial cells that surrounds an empty or fluid-filled lumen. And so if you then zoom in on that material and imagine what that straight tube or flat sheet of epithelial cells has to do in order to deform into a branch or into a cul-de-sac, there are three possible mechanisms, right? The first is that that material actively folds. The second is that it passively folds because it's being pushed from its apical surface, the side that faces the lumen of the airway. And the third is that the epithelium passively deforms because it's being pushed from its basal surface, the surrounding stroma or the mesenchyme. So we've been using the vertebrate lung to try to uncover the mechanical forces that turn really, really simple epithelial tissues, sheets and tubes, into complex structures, and taking advantage of several different species, shown here, as models of anatomical, physiological, and mechanical diversity. So what I'm going to do today is to summarize a bunch of work, first in chickens, and then in lizards, and then in mice, and then use that as a backdrop to try to reconcile some of the differences between epithelial morphogenesis in this organ that is common amongst these different classes of vertebrates. So I'll start with the chicken as a modeled bird. The bird lung is parabronchial, which means that it exhibits unidirectional airflow when it's mature in the adult, but the initial structure is fairly simple. The lung begins as an epithelium that is wishbone shaped, indicated here in orange, and then it ramifies, so forms new branches by generating branches off of the dorsal surface in really highly stereotyped locations during embryonic development. And using the chicken as our model system, we found that the epithelial cells that give rise to branches do so by undergoing an active change in cell shape, an apical constriction, such that they shrink their apical surfaces, which are adjacent to the lumen, and they expand their basal surfaces, which are adjacent to the mesenchyme. And this local change in shape is sufficient to bend the epithelial tube into a branch in that location. And we can model that process computationally by specifying patterns of apical constriction on a thick-walled cylinder with the same material properties that we see in the embryonic bird lung, and then applying the principle of finite volumetric growth. So for the lungs of birds, including chicks as well as other species we've investigated like quails and ducks, we found that the epithelium actively folds itself into new branches, and right now we're really excited to use engineering approaches to try to fold tissues by manipulating patterns of apical constriction. More recently, we started investigating the physical forces that drive the morphogenesis of the reptile lung, which is shown here in cartoon form on the right, and then a relief map in this rainbow coloring on the far right. The reptile lung is essentially a hollow bag with no branching, but with a really bumpy epithelial surface of faveoli that promote gas exchange, really similar to the alveoli in our lungs. We started just before the pandemic hit by establishing the brown anole lizard as our model organism. It's easy to maintain in captivity. It's a highly invasive species. Its incubation time is about a month, and the adult is about the size of a pencil. So it's a fairly tractable model organism. And what we found is a completely different physical mechanism for development than in the bird lung. So here the lung begins as a simple epithelium. I'm showing you one side in the top row here. And then in green in the bottom row, so either the left or the right lung on the far top left image. So an initially smooth epithelium that is covered uniformly along its basal surface in another tissue called smooth muscle, which is shown in the center row here, and then in magenta in the bottom. And over the course of 2 days, so moving from left to right in these images, that initially uniform smooth muscle tissue migrates to form a hexagonal mesh through which the epithelium actually protrudes in response to pressurization of the fluid within the lumen of the organ. So we tested that concept several different experimental ways. We can also model the physical mechanism itself. And that model predicts the change in morphology that we see. So our model consists of both the epithelium, shown in green, the smooth muscle mesh in purple, and the pressurized lumen. And what we find is that increasing the pressure of the fluid in the lumen in the model is sufficient to push the epithelium, which is a very quiescent tissue at this stage of development, through the holes in the mesh. Right, so we call this process stress ball morphogenesis because it actually resembles the toy that you might have on your desk, where when you squeeze the toy, you increase the pressure, and that leads to expansion of the elastic tissue, if you will, through the holes in the mesh. All right, so the reptile lung builds itself using a completely different set of forces than the bird lung does. And specifically, it's a passive surface that's sculpted by forces from the lumen of the developing organ. So with that as a backdrop, I want to spend the rest of my time sharing work focused on uncovering the mechanical forces and signaling pathways that are responsible for controlling morphogenesis of the airways of the mammalian lung, using the mouse as our model organism. In the mouse, the lung begins also as a simple tube, the trachea, that splits into 2 primary bronchi that then recursively branch to build the 8 million tubes that are present in the adult organ. And at the same time as that epithelium is undergoing this branching morphogenesis process, the surrounding mesenchyme is differentiating into airway smooth muscle, which is shown here in red, which wraps circumferentially around the epithelium. What we found is that there's no evidence for apical constriction in the developing embryonic mouse lung, and instead, we found that this smooth muscle differentiation plays a really important role. So using transgenic mice that express red fluorescent protein downstream of the alpha smooth muscle actin gene promoter, which fluorescently labels all smooth muscle in the developing lung, we found that airway smooth muscle first appears at the basal surface of the epithelium at the future branch site before the epithelium changes shape, which I hope you can appreciate by seeing this white spot appear at the top of the epithelium in the center image, in the center movie. And then that smooth muscle wraps around the nascent branch. And so again, we can model this computationally. What we found is that those smooth muscle cells that are differentiating are stiffer than both the epithelium as well as the rest of the mesenchyme. And that pattern of differentiation of a slightly stiffer tissue from a soft mesenchyme is sufficient to change the shape of the growing epithelium. Which we found both experimentally, and I'm showing you modeled here computationally, with the growing organ as 3 different layers of tissue: the epithelium in green, the smooth muscle in purple, and the undifferentiated mesenchyme in gray. Okay, so all of our data suggests that this pattern of smooth muscle differentiation serves as a mechanical template to constrain or serve as a girdle to shape the proliferating and expanding epithelium into branches. And that means, of course, that there are 8 million patterned events of smooth muscle differentiation in the developing mouse lung, and we're now investigating how this is controlled. Okay, so all of our data suggest collectively that the mouse airway epithelium is folded passively in response to stiffening of the local mesenchyme as it differentiates into this new tissue layer, the smooth muscle. And we're now poised to ask how mechanical forces actually are transmitted into the mesenchyme to help to control or specify that pattern of smooth muscle differentiation. All right, so that physical mechanism for driving morphogenesis is distinct. It's very different from what we found in birds and what we found in reptiles. And so we've therefore revealed at least 3 different physical mechanisms by which the lung epithelium is deformed during development. And these distinct mechanisms are despite the fact that the signaling pathways that I mentioned at the very beginning are conserved across mammals, birds, and reptiles. So these exact same signaling pathways have been identified in all three different classes of vertebrate lung morphogenesis. We have very different physical mechanisms that result from that signaling. So I want to finish in the last couple of minutes by discussing why the physical mechanisms aren't conserved, which is a finding that is surprising and can be rather distressing to think about, given how much we as a community collectively value conservation. Right, so why aren't the physical mechanisms conserved despite the fact that the molecular signaling pathways are? Well, I started this discussion today by using an analogy, right? I said that the tissue that folds is like the wall of a balloon. And if you consider that analogy even further, then just focus on that balloon and think about how easy it is to expand or change the shape of the wall of any balloon, right? It really depends on the properties of the wall itself, right? So a regular old party balloon is a lot easier for you to blow up than a punch ball balloon that you might buy for your kid that has a really, really thick ball, right? And so the properties, the mechanical properties, the material properties of that balloon affect its ability to deform. And similarly, the epithelium is very different at the earliest stages of development in these three different classes of vertebrates. In birds, the tissue is columnar. It's really, really thick. It kind of looks like an early stage Drosophila embryo. In lizards, in contrast, the epithelium starts out cuboidal and then it becomes really, really thin and squamous as the pressure increases within the lumen of the developing organ. And in mice, the tissue is somewhat in between. It's pseudostratified, right? So despite the fact that the signaling pathways are conserved, the initial physical conditions are different here. In the case of the bird, we have thick, rigid tissue that has to fold itself. And in the case of reptiles and mammals, we have thin, floppy tissues that expand and fold in response to external forces. So this is somewhat similar to the geometry argument that Tomas was making last night, except it's more than just the geometry around the tissue. It's the geometry and the resulting mechanics of the tissue itself. I also said that we get our final structure from folding those initial starting materials. And here I think it's quite revealing that the initial conditions, or that the final conditions, across these different classes of vertebrates are also quite divergent. So birds have lungs that are made of thick, rigid tubes. The lungs themselves never inflate or deflate like ours do. And in contrast, our lungs, mammalian lungs, and reptiles have really compliant lungs whose volumes change in response to pressure. So we have different initial conditions, different final conditions, and different mechanical routes to achieving them. Right, so I'll pause it. I'll leave you with this, that a big challenge for the field in general and for 4DCP, probably in particular, is taking this really sophisticated gene expression and imaging approaches that have been developed recently and that have given us cartoons like the one on this slide, and creating similarly high-tech, highly sophisticated means for understanding how the material properties of these systems change over space and time. I think without this understanding of mechanics, then our parts lists will remain, frankly, just that. So my group in particular is really excited to continue working in this area and defining the physical forces that govern morphogenesis, and especially in looking at other species to uncover different routes that are used to build tissues. So our zoo is ever expanding. All the work that I showed you was generated by a fantastic team. I summarized quite a lot of work. And with generous support from our funding agencies. And with that, thank you so much for your attention and for your understanding. And I'd be happy to take any questions.
Really nice talk. In human lung development, for example, surfactants are super important for decreasing surface tension. What happens in birds and in reptiles? Do they have surfactant, and is it different?
Yeah, that's a fantastic question, right? So if you're going to inflate or deflate something that's on the length scale of about 200 micrometers, which is the length scale of our alveoli, then surfactant becomes really, really important for being able to maintain patency. We've just started to carry out some single-cell RNA sequencing analysis of embryonic lungs at multiple stages of development in chicks, in anoles, and as well as some in other species, like we're getting started with tadpoles, actually, to pull out the similarities and differences that one might see, both with respect to the tissue types that are present, the cell types that are present, but also to get at this question that you're asking about, the role of the non-cellular compartment, like surfactant in terms of specifying the mechanical properties, the material properties of the tissues. I mean, I can tell you that the reptile lung does appear to have surfactant, and that would make sense because it's highly compliant, like ours are. It's a little bit unclear what surfactant would be doing in the case of the very rigid bird lung. So the airways don't expand or collapse during airflow in the bird. But if we find some conservation of surfactant proteins in the epithelium of those airways, it'll be really interesting, I think, physically to understand what it might be doing there.
Great. Temperature plays a role in physical properties of materials, right? And I see that the structure of these lungs are different. Is body temperature playing a role in the architecture of the lungs development?
That's a wonderful question. So what's the role of temperature here? Temperature is fairly well defined in the case of a mammal, right? So we develop in utero, and so the temperature is controlled largely by the mother. The developing bird embryo will not continue to develop its lungs unless it's brought to a fairly similar temperature, so around 37, 38 degrees Celsius. So we don't really have a lot of ability to tune temperature in those two classes of vertebrates. I think where temperature becomes probably a little bit more interesting is trying to understand some of the diversity of timing of rates, as well as physical mechanisms in reptiles, where you have a much more diverse set of species, really diverse set of final architectures. So the anole lung that I showed you is probably one of the most simple. There are much more complex reptile lungs out there that we're really excited to explore, and we're taking advantage of the veiled chameleon as one model system to get there. But what's really cool about reptile development is that it is tuned exquisitely by the temperature of the surrounding environment. And the duration of incubation for development of many of these species can be an order of magnitude different, despite the fact that they're about the same size. And so I think that's where we'll probably see interesting effects of temperature, specifically in this class of vertebrates, the reptiles. But with respect to material properties, I don't think we're going to be able to pull out anything in mammals or birds where temperature is really, really tightly controlled.
I'd like to jump in with a question from online from Alan K. Does transepithelial water movement driven by the sodium pump have any role in the development of alveoli?
Okay. Probably. We do see that there is an increase in fluid volume in the development of both the mammalian lung as well as the reptile lung that I showed you an example of. There, the increase in fluid, the dynamics with which that fluid builds up differ between those two different species. In the case of the mammal, a lot of the transport of fluid into the lumen does appear to be from ion transport across the epithelium itself. And so the answer to your question is probably. The specifics, I think, will really depend on which stage of development one is looking at and which change in morphology of the epithelium one is trying to consider.
Quick one from Xiaohei.
All right, I actually have several, but I will just ask one. So really cool work, Celeste. I also want to say like I really want to talk with you in person, but we can talk about that later. So have you looked at the dynamics of the cells in the reptile lung and the bird lung? Are those kind of like, if I think about temperature, I often think about the dynamics of the cells as the tissue's temperature itself, right? So is that also influencing the mechanical properties, like how fluid how deformable they are?
Yeah, Shava, thank you for the question. I'm also sad that I'm not there in person to chat with you. I really, really tried hard, y'all, but I can't drive right now. So we've looked at tissue dynamics, we've looked at cell dynamics. Where we've seen the most interesting changes in cell positions with respect to their neighbors actually is in the mesenchyme. And so I was, really inspired by Amy's talk yesterday. The mesenchyme is kind of like the ugly stepsister of tissues. Everyone focuses on the epithelium because that's the business end of the organ, and that's what appears to be doing the interesting stuff with folding into branches and building beautiful bulges on the surface of the skin. But we've seen the most interesting dynamics in the mesenchyme. Actually, we don't see a lot of neighbor exchanges in the time-lapse movies that we've acquired for developing lizard lungs. We don't see much at all with respect to changes in neighbor positioning in the bird lung, and I think that makes sense because they're all kind of like really tall columnar epithelial cells packed adjacent to each other. But we do see a lot of dynamics within the mesenchyme. So an increase in fluidity, particularly in the bird lung, as the epithelium bends and pushes into the mesenchyme. And we think that what that does is to decrease the resistance to expansion and extension of the branch itself. But not a lot with respect to what's going on in the epithelium. And so here's where I think like having, sort of a broader tissue kind of viewpoint that I think can, really benefit from all of the tools that have been pioneered and developed at Janelia, can get us more understanding of the physical processes of morphogenesis of these and as well as other organs as well.
All right, let's thank Celeste one more time and wishing you the best for your ribs. So now I have the pleasure of introducing Zeb Gartner from UCSF. He's going to tell us about building tissues to understand how tissues build themselves.
All right. Thank you. And just to ditto what's been said many times before, Janine, thanks so much for putting on such an awesome meeting, you and your team, and to Jennifer and Ron for injecting their leadership and intellectual power and everything into this field. It's exciting to be here at the beginning. So my lab at UCSF is really interested in tissue structure. And so we ask questions similar to what many people in the last couple of days have been talking about, which is how does tissue structure form during development? How is it maintained during homeostasis? And we're interested in the answers to these questions for purely intellectual reasons, but also because they can have really important applications. So, for example, if we were better able to control the composition or the state of community of lead to these changes in structure, we can imagine going in and perturbing those pathways, stabilizing tissue architecture, to test the idea that maybe that would actually block disease progression. And finally, from a purely tissue engineering perspective, if we can learn what really guides the formation of more complex and functionally mature tissues in a dish, that would give us the possibility of building better models of human disease, better platforms for drug screening, and I think most intriguingly, new replacement parts for the human body, for regenerative medicine. So towards thinking about these questions and goals, we begin by sort of defining what we mean by tissue structure at a very high level. So I think Ruslan already sort of mentioned some of these metrics. So we think a lot about the size of a tissue, so the number of cells that are there, the composition of the tissue with respect to the cell types and the states of the cells that are there, and also the shapes of the cells and their three-dimensional arrangements. And so the tissue I want to talk about today is the mammary gland, but hopefully you'll see that the concepts are hopefully generalizable here. But the mammary gland is a tissue tasked with milk production and milk secretion during breastfeeding. It's a branch ductal organ. If you look at cross-section through the epithelium, you get a structure sort of similar to what I'm showing here in the upper right. So there's two compartments to this tissue. There's what's called the luminal compartment, comprises two lineages of cells shown in green that I'm just going to call luminal epithelial cells. And among other things, they're tasked with milk production. And they're surrounded and separated from the basement membrane, which is shown here in blue, by another population of cells called myoepithelial cells. And these cells are contractile. And again, among other functions of these cells, they're tasked with contracting to eject milk during breastfeeding. So these sort of textbook diagrams of what tissues look like tell you a lot about sort of the average structure of a tissue and the sort of the basic design principles. But as I think most people here would recognize, if you actually go in and look at what these tissues look like, say, for example, in tissue sections, it looks a lot different. So here's a cross-section of the human mammary gland. And what you see when you look at this, just qualitatively, is that these three metrics of tissue structure that I described are incredibly heterogeneous in these images, right? So we see ducts of different sizes. You can't really see it from this image, but I'll show you in a second. They're very different in their composition. And also the arrangement of cells can be different. You know, occasionally you see these keratin 14 positive cells. These are the myoepithelial cells that are a little bit in the wrong place, right? So this really began to crystallize for us when we went in and started to quantify these properties of tissue in real human specimens. So this was a study from Lindsay Murrow just this year where, among other things, she went in and asked, how does the composition of the mammary epithelium differ among individuals? And so she did this by immunofluorescence in situ, she did by flow cytometry, and this data here is single-cell RNA sequencing. And what hopefully is like painfully obvious looking at this is that, man, these tissues are incredibly heterogeneous. So, you know, what gives? Vasudev Srivastava in the lab went in and looked at a different cohort of patients to look at the arrangement of these cells. So I mentioned this one structural metric. You know, myoepithelial cells separate the luminal cells from the basement membrane. So she asked, what fraction of luminal epithelial cells across ducts and acini in this tissue actually come in contact with the basement membrane? And just looking at one patient's tissues, this is what she saw. So again, it's like it's all over the place, right? And so these are, you know, static views of the heterogeneity of tissues. And anyone that's worked with organoids knows that the same sort of properties of heterogeneity are evident in engineered tissues as well. But it's also worth emphasizing that these tissues are heterogeneous in time as well. So we all recognize this through the lens of development, where tissues change structure. The mammary gland is interesting because it actually goes through development after birth, postnatally. So during puberty, during pregnancy, lactation, involution, and also during every menstrual cycle, right? But those are sort of developmental timescales. If we zoom in and look at much shorter timescales, say tens of minutes to hours, these tissues are also highly dynamic. So this was a study from about 5 years ago that used intravital microscopy to look at cell dynamics at two anatomical locations in the developing mammary gland. So out at the end, the terminal end buds, where the tissue's actively invading into the surrounding stroma, and further down on the ducts, where the sort of tissue structure had already been established. And so looking at this top panel, what the authors did here is they outlined the cells in white. And so what hopefully is evident is that cell shape is highly dynamic, as well as the topology of cells, so who are their nearest neighbors. Whereas if you look at the same mammary gland, but further down on the ducts where tissue structure has been established, the dynamics looking totally different, right? So cell shape is much more fixed, as well as topology. And what I think has become increasingly evident, and I think is really exciting, is that all these properties of tissues, so their structure, their heterogeneity, their dynamics, seem to be fundamental properties of the cells themselves and their interactions. So they emerge from those. And we know this because you can take these tissues out of the body, separate them into single cells, reconstitute them back together, and they recapitulate many of these same properties in a dish. So this is a movie from Andy Ewald's lab, just showing how on the right, this invading tip of the organoid has these really elevated cell dynamics. But looking out at the walls, although you still see cell dynamics, they're considerably reduced. So hopefully I've convinced you that these are sort of fundamental properties of tissues. And we really wanted to begin to ask, what are the drivers of this heterogeneity, really at a fundamental level, right? And so you can imagine trying to tackle this problem in vivo in the most physiologically relevant context. And here the problem would be that we already know that these tissues are incredibly diverse in their sizes, their compositions, and cell arrangements. And I think intuitively, we all know that these features of tissues are somehow linked to one another, right? So just to give you an example, if we completely got rid of myoepithelial cells, all the luminal cells are going to be in contact with the basement membrane. So these things are in contact, associated with one another. But I think more broadly in vivo, there's a lot of microenvironmental heterogeneity. People already know this. And it remains very difficult to image dynamics, at least in a sort of a complete way. So the strategy that we've been taking, and I think we've already heard from Xiaowei and Amy about how powerful this approach can be, is to take a reconstitution approach. So what we can do is use sources of tissue like breast reduction, mammoplasty tissue, and we can dissociate this down to single cells, and we can use techniques like flow cytometry to isolate the various lineages to near purity. And then we can take advantage of a whole variety of bioengineering techniques pioneered by people like Celeste to bring these cells back together, but under highly controlled conditions where we can control the size of the tissues, we can control their composition. We can culture them in a common microenvironment like Matrigel, despite its problems, it's still pretty good. And we can use techniques like fluorescence microscopy to image their dynamics. And so the movie on the right is just a representative, actually it's not really representative, it's a really nice movie of the self-organization of one of these tissues. All right, so this is an approach that two just really, really incredible scientists in the lab decided to take, Dr. Vasudha Srivastava and now Dr. Jennifer Hu. They used this strategy to basically make a gallery of many hundreds, thousands of these tissues. And so this is just looking at a few of them, which approach a steady state after about 24 hours. And so what hopefully you can take away from these videos is that these cells remain incredibly dynamic. They're changing positions. And the myoepithelial cells are in general enriched on the outside of the tissue. But we wanted to ask, can we quantify this? So they put together an image analysis pipeline that allowed them to segment these images and then extract out this one specific structural metric. So what fraction of these green luminal cells are touching the edge of the tissue, the basement membrane? Aggregating all this data, what you get is a probability density function that looks like this. All right? So what can we take away just from this? Well, one, there's a most likely configuration of the tissue, and that's one with about 20 to 30% of luminal cells touching the basement membrane, which is encouraging because it's similar to the heterogeneity that we see in vivo. Interestingly, we do see a small fraction of tissues that have this textbook architecture with no luminal cells at the edge of the tissue. But also interesting, we essentially see no tissues where all of the edge of the tissue is occupied by luminal epithelial cells. So clearly there's order in this distribution. So we built these tissues with 50% green and red cells, but we see on average more red cells, more of these myoepithelial cells in the tissue edge. But also it's notable that this distribution is shifted to the right from what you would expect from this textbook image, and it's also really broad, right? So looking at this, the questions that we began to ask were, what were the processes that were pushing order, imposing order on these cell configurations? And similarly, what were the processes opposing that were promoting disorder at this tissue scale? So cell state, and cell identity at least in the mammary gland, is fixed on the timescales of these experiments. So we focused in on the mechanics. So about 70 years of research has really pointed to the mechanisms that drive these active rearrangements of cells and tissues, which is called cell sorting. And the way that the modern view of how we think about this is that at the interfaces between every flavor of cell in the tissue and the ECM, these different lineages maintain lineage-specific interfacial tensions, right? And so these represent forces that cause some interfaces to expand and other interfaces to shrink. And so we can begin to think about an overall surface energy of a tissue like this as being the product of each flavor of surface area in the tissue times its interfacial specific tension, right? So summing this all up, you get a total sort of mechanical potential for the tissue that you might imagine. And so what the tissue is sort of trying to do is sample around in configuration space to converge on a low energy configuration. So we can ask, is this contributing to the tissue and how? So we can use techniques like micropipette aspiration, Young's equation. We can measure all of these interface-specific tensions. And just at a high level, what's really guiding formation of this tissue is that myoepithelial cells have a very favorable interaction with the ECM, explaining why they're enriched there. Luminal epithelial cells have a very unfavorable interaction with the ECM, explaining why they're excluded from that position. So this is nice. And so we can really write down what the mechanical potential for this tissue looks like, and that's shown on the right. So it's a monotonically decreasing function of the proportion of luminal epithelial cells at the tissue edge. But there's a problem here, is that this was the full picture. What we would expect is that all the tissues would have that beautiful stereotype textbook image, and that's not what we see, right? So something is actively promoting disorder here, if this is true. So we spent a lot of time scratching our heads about this until Vasudha had a really beautiful insight, and that's that she reconfigured in her mind this tissue as sort of a two-compartment system. There's edge sites and there's core sites. And she asked, if I just started taking red and green cells and randomly dropping them into these lattice sites, what would be the baseline probability of observing different configurations? And so when you do this, it turns out that it's incredibly likely that you see the configurations at the top, which look disordered, right? It's incredibly unlikely that you see the configurations at the bottom, which are highly ordered. There's only a few ways to put together those building blocks to get those configurations. And she wanted to ask, could we actually measure this baseline probability of observing the different configurations of this tissue? So one way to do this would be to take luminal and myoepithelial cells and carefully engineer them so that there's no more mechanical potential. That's hard. Instead, what she realized is that she could take two populations of myoepithelial cells, or luminal cells, and just give them different labels. You've completely gotten rid of the mechanical potential now, and so you can mix them back together and build them back into tissues of the identical size, composition, and everything else. They remain dynamic. They continue to sample from their environment, and when you measure at the steady state the distribution of configurations, now it's nicely centered at 50%, which is the composition of the tissue, and it's beautifully fit by a Gaussian distribution. Right? So the question then becomes, how do we combine our knowledge of this mechanical potential with the baseline probability of seeing different configurations of the tissue to predict what this distribution should look like that I'm showing you here in blue, right? And luckily, we can take advantage of tools developed from statistical mechanics to build a very simple model for doing this. So what Vasudha asked is, could she basically model this distribution using Boltzmann statistics? So she reimagined what this distribution was, and she defined this term, the probability of your ith macrostate. So that represents each of these bins in this probability distribution. And she parameterized this expression using a couple measurable features of the system. So the first is this term omega, which represents the degeneracy at baseline of each of these macrostates. And so this is measurable, and it's the gray curve that I'm showing here. We also have this term E sub I. This is the average interfacial energy of each of these macrostates, and I already showed you how we can measure that as well. There's another term in here, beta, and so this is the inverse of activity. And so as Xiaowei pointed out, you can think of this as being similar to an intrinsic tissue temperature. Mechanistically, what this represents is fluctuations in the mechanical potential of the cell surface around an average value, which is what we measure. This is critical for cells being able to move around. And finally, we normalize this probability distribution to have an area under the curve of 1 using the partition function. Right? So this very simple expression has really surprising predictive power, and we can use this now to begin to engineer this ensemble distribution of tissues in the dish. So just to give you some simple examples, if you hold cell identity fixed and the size of the tissue fixed, what this model predicts is that if you increase the proportion of luminal epithelial cells in the tissue, you should broaden the distribution and shift it to the right. And so when we test this, the data strongly supports the predictions of the model. Similarly, if you hold the composition of the tissue fixed, as well as cell identity but vary size, you would predict that you would shift the distribution to the left and narrow it. And yet again, we see nice concordance between the model and the experiment. Finally, we can go back to the tissue that gives rise to the gray distribution, where it's all myoepithelial cells, and manipulate specifically the mechanical potential by going in and knocking down talin-1 in one of the two populations of myoepithelial cells. So this effectively decreases the affinity of those cells from the basement membrane. We can measure the mechanical properties of the cells, and what the theory again predicts is that we'll shift this distribution to the left, and again, that's what we see. All right. All right, so I'm going to just wrap up really quick and say that this very simple conceptualization of self-organization provides real predictive power from an engineering perspective. I think at a fundamental level, what it's pointing out is that tissues are constantly fighting between order and disorder, and that they can actively modulate this balance, for example, by using activity. But what this is really telling us is that there's an effective limit to the order that one can impose on a tissue in dynamic processes. Looking forward, we're really interested in thinking about other driving forces for order. So for example, cell-cell signaling, thinking about morphogens. We're really interested in how other organisms like C. elegans are so precise, they're able to just narrow this distribution down to effectively a delta function. And the thing that we're working most keenly on right now is really trying to think about what this teaches us about cancer, right? So how does disorder increase under those conditions? So thank you very much for your attention. I just want to thank the current and past members of the lab and our wonderful collaborators shown at the top and our funders. So thanks.
Canalize the distribution, change the statistics, would be if there was a way to, for the ensemble of cells to actually tune or self-organize a proportion of the various cell types, as opposed to it being, a pre-pattern that you experimentally introduce. Do you have any sign that in some conditions that the proportions could be self-regulated, and therefore the system would actually directly go to a narrow distribution or a broader one? I don't know if it makes sense.
Yeah, no, I think I get the gist of the question. It's interesting that during development, I think there's an emerging body of evidence to suggest that morphogen gradients may be doing as much to specify composition as they are position, and that some of these mechanical processes sort of allow an initially sort of coarse distribution of cells to be refined in space. In this particular example, we were imposing composition on the tissue and sort of forcing it to be fixed. But how that's established in the mammary gland, I think, is a really interesting question, right? So you do have two different compartments, and I think it's a reasonable hypothesis to say that cells differentiate into a state that's consistent with the compartment that they're in. But whether that actually occurs here, surprisingly, is something that hasn't really been tested, and so I think it's something that we should look at.
That's a super cool talk. I was wondering if I can get a clarification and then a small question. What I understood was, in theory, you should see this symmetric distribution of entropic states, but in your observed data, there's a particular skew such that in theory, the rare states should be symmetric, but then you see this shift towards the left-hand side, right? Mm-hmm. And then that seems to suggest that the composition of the cell rather than the radius is the most predominant factor in driving that skew. Is that correct?
So at least in the cases that I showed here, the skew, all these things are interconnected, right? So you have to hold two parameters fixed to understand the impact of the third, right? When we hold composition fixed and size fixed, the skew comes entirely from the mechanical potential, right? But I also showed an example where we hold the mechanical potential fixed and the composition fixed, and the skew can be biased by the size of the tissue. The reason for that is because as you scale the tissue, the number of surface sites to volume sites change, and so the impact of entropy on the distribution goes up.
I see. And the quick question is, this is a two-state model, how do you imagine this to scale to a multicellular?
Yeah, I mean, that's super exciting. I mean, I think that as you build more complexity in the tissue, there's no fundamental reason why you couldn't make a more complex version of this model. And I think another interesting thing to think about here is for other developmental processes or processes that occur at the steady state in adulthood, there are other mechanical fluctuations that give rise to tissue structure. For example, the buckling processes that were described by Celeste, the mesenchymal patterning processes described by Amy. When you can articulate a mechanical potential and you can define the states of the system, at least in principle, you could apply this class of model to it to try to understand what's going on.
Thank
you. Zeb, that was fabulous. I mean, this beta term, the fluctuation term, I mean, that's coming from activity in the system. Mm-hmm. And is that I mean, how does that scale with the different types of structures and organization that you have? I mean, and does it work out to what you can measure in terms of the fluctuations of the two cells themselves? I mean, or is it measurable, or is the parametric fit?
Yes, I think this is a really fundamental question, and one of the really important things about activity with respect to how we could use this model to sort of find deeper insights into tissues sort of at the bulk scale is that not only do we need to understand what it is, but we need to understand how to manipulate it independent of, say, the mechanical potential. And that's been a very vexing problem to us. So what we've been doing thus far is trying to infer what the activity should be by measuring other things. So our favorite measure thus far has been to label nuclei of cells and measure the mean squared displacement versus time. So it's like diffusion, but that's a function of temperature, right? I can show you one interesting experiment that I think shows a signature of this. So we can measure activity by taking two aggregates and allowing them to come into contact, and then measure the time that it takes for them to fully mix. So this is what happens when we put two aggregates together in Matrigel. So they fully mix in about an hour or so. We found a microenvironmental condition that allows us to manipulate activity, and that's taking these aggregates and putting them into agarose, right? The reason we think this works is because activity seems to be injected in the tissue primarily through the motion of the myoepithelial cells that move on the ECM. So when we remove the ECM, the activity of the whole tissue drops. So this is sort of what agarose looks like, the curve in orange, right? The reason this is interesting is because it also allows us to test the impact of modulating activity on tissue structure. So we've been able to identify conditions, for example, not by manipulating talin in these cells, but by manipulating cell-cell adhesion by knocking down P120. This also generates an energy gradient in the tissue, right? But it's very shallow compared to that of talin. So what's really interesting about this is when you look at these tissues' ability to self-organize in Matrigel, they essentially don't organize at all, right? But you take those same cells and you put them into agarose, you're dropping down the activity, and now you get order imposed on the tissue again. So, what's the surgical perturbation? Like, we don't know. Like, what is it that we should be measuring? It's hard to say. We don't have that thermometer, but this is super key to like moving this forward. Yeah.
All right, let's thank Zeb one more time. So now it's a discussion, and it's going to be moderated by Zeb and Amy Shire.
Yeah. Amy's here. I'm going to get my notes.
So we're going to do like we did yesterday, and also open up the chat to our virtual audience members and project the chat on screen shortly. So Zeb and Amy, maybe you guys can sort of just keep an eye on if anything comes in from the chat too.
I'll give you this to pick around.
How do we do that? It'll come up there on the.
Oh.
It'll come up. It'll be up in a minute. And Celeste is also joining us backstage.
Hi, Celeste. Okay.
Go ahead.
Okay, I'll start the discussion by asking your opinions on cell-based modeling versus continuous modeling. So what's your opinions on, to me, as a cell biologist, I just feel the most sensible thing is to treat cells as cells and then use that as a unit for the modeling. But I also see the, like advantage of using some of the continuous-based modeling to get some intuition. So how do you reconcile being too simple versus simple enough?
To get started?
Sure. Yeah, we were going to be asking the questions. We'll answer
Yeah,
them
come on,
too.
shall we? We were supposed to be asking the questions. I mean, I can maybe start. And, so, I had an interesting conversation about this the other day with somebody, and one of the takeaway messages for me was that one of the powers of continuum models, at least, is that if you can write down an analytical expression, you can really explore the space of the parameters on the system to ask questions like, what are the attractors? What are the low energy configurations, depending upon the class of model? But you really need a concrete theory, mathematical expression to do that. And I think one of the nice things about the cell-based models is that although they can sometimes seem a little bit ad hoc, you can very quickly test your intuition in them by changing some of the parameters and sort of seeing the way the system changes. And so it could be really powerful, at least early on in a field or a system, in terms of testing your intuition and designing new experiments.
Yeah, I guess I would add that I think in my experience, there are times when the relationship between the cells is what you really want to be considering, and then there are times where sort of the properties of the whole are maybe what's most regulatory in a particular moment. So I think it's not necessarily that one is simpler than the other, but just sort of scale dependence, I think, plays a role. Others can feel free to comment as well.
Celeste, anything to add?
Yeah, I agree with that last point. I think for our systems, it really depends on the length scale and the time scale of the process that you're interested in. Like if something is happening on a large enough length scale where the business end of the morphogenetic event is the tissue, then a continuum model will give you a good enough approximation that you can design experiments then to test your hypotheses based on predictions from the model. If, however, you're interested in events that are happening more at the cell scale, like Shava's question before about cells rearranging with respect to each other, then a continuum approximation isn't really going to permit you to design an experimentally tractable, predictable kind of test. So it just really depends. They both have their utility, I think.
Well, Zev and I were thinking about opening the discussion with maybe an even broader question, which is just that if we're working under this heading of mechanistic tissue biology, going forward, how do we as a community want to define mechanism? So I thought, given especially sort of the multi-scale nature of what we're thinking about, how do we want to think about really that word?
So I don't know what order we go. Can I go?
Please.
I have a strange question, actually, induced by Zev's talk. Can you think of, but then it's broader to all of you, can you think of a way to make that process of self-organization that you described reversible? What would you need to change to reverse it? And the reason I'm asking about it, one is other particular parameters would be more sensitive, but reversible, not necessarily, going backwards, but going from this organized structure to alternative structure that would be you lose what was developed by the process, and you reverse to initial state, which doesn't mean you go through the same trajectory. The reason I'm asking that, one is just from theoretical perspective, it's interesting what would be the sensitive parameters, but some tissues have reversible remodeling, like breast, right? After weaning, this process reverse. Again, they don't go the same path as they went during pregnancy, but they are reversed nevertheless. The same is neurogenesis, synapses are formed and extra synapses are pruned. And I think that this reversal of some of developmental programs, maybe not studied as much, but they're maybe clinically very important. That's where, like, particularly I think in lung and the skin, or in conditions like systemic sclerosis, where I think inappropriate reversal of branching morphogenesis may be actually underlying culprit. So I'm just wondering what people think about going in reverse in development.
Yeah, it's a really interesting question. And you know, when you talk to cancer biologists, for example, it's sort of tissue morphogenesis in reverse. You know, you're losing some of the organization of the tissue. I guess what we would predict, I think it might be simplest to answer that question by focusing on a single tissue. So like in the mammary gland, there's two ways to sort of go in, or at least three ways to go in reverse, right? So in the mammary gland, early in development, there's only really a basal-like cell type. And so it's specified, like the luminal cell type is specified by actually shutting down a significant region of its transcription through rearrangement of the chromatin. So you could imagine changing the composition of the tissue by going more basal, right? So that's one way of going backwards. Another way of going backwards is to think about going from a static tissue, like say a duct, to something like a developing terminal end bud where you have a lot of activity, right? And I showed you an example of like where you can take a tissue which is organized, and you increase the activity and it disorganizes, right? Even though the mechanical potential is still there. And a third way of doing it is holding all that stuff fixed and changing the mechanical properties of the cells themselves. So for example, if a luminal cell began to increase its ability to interact with the ECM, right, that's going to shrink that mechanical potential or make it more shallow. And so the tissue would kind of go in reverse. So I think it's tempting to think that the similar things happen in other tissues, but I think in order to answer that question, you sort of need a detailed understanding of not just the development of that tissue, but what are the sort of, as you say, the sensitivity of the different parameters there.
Yeah.
Ron has a question too.
Oh, yeah, Ron. Okay.
I'll ask one. I had one question, and this is related to what is a mechanistic understanding. So basically, can we connect like cellular behaviors, particularly developing tissue environment, with the intracellular signaling events like reporters? I mean, I'm particularly referring like tool development, sensor development for all those motility-related pathways and their applications in tissue environment. Can you comment on the current difficulties, what we need, and what kind of information are you expecting from that? Thanks.
Yeah, I'm in full agreement that some sort of cell-level sensor of what's happening intracellularly on these mechanical fronts would be a huge breakthrough for the field. And these sorts of tools I really envy in contexts like cellular metabolism. And so maybe this sort of world would allow us to better see what commonalities and maybe build bridges on the tool front so that we could import these types of sensors into measurements of what's happening mechanically. But I would agree that that's a real bottleneck, and open to ideas or suggestions for how to broaden that toolkit.
Well, maybe I'll also bring Celeste and Tomas into the conversation here too. And maybe this gets, partly a mechanism, although different people have different views of mechanism. But we hear these beautiful talks of these individual systems, and I'm kind of hearkening back to what Ruslan brought up. There's a billion specializations of general mechanisms, and of course, people in their individual lab, if you're a postdoc or a grad student, you're working on this problem that's right in front of you with your system and trying to figure out how it works. But we've heard now common themes here. We've heard about contractility through myosin. We've heard about geometry and how the container boundary, affects what you can create. We've heard about alignment of the ECM. We've heard about other factors like adhesion. How far is one away as a community now to look at this as a toolbox for creating a plethora of biological shapes, but with a limited number of rules. I mean, that's not just the challenge to this whole group here, maybe to invoke. I mean, are we close to that? Very far away? Are we not talking across systems right now? What needs to happen, to, get at, principles of tissue shape?
I think we're already there. I mean, we have a huge toolbox of physical mechanisms by which cells within tissues can construct elaborate forms. I think the disconnect is understanding how all of those signaling pathways, I keep thinking back to Hernan's beautiful example of the early Drosophila embryo. So how do all of those signaling pathways impinge upon many of these mechanisms like ECM alignment and cell contractility? I think there's a disconnect between those two languages that needs to be filled in so that we can start to apply, the rich diversity of signaling and gene expression that's already been uncovered to these more physically minded kind of tools that are present within this toolbox that is being constructed. But to answer your question, I think we're there with respect to having a large understanding of the tools that are used by nature to build complex morphologies.
Well, I'll just extend that as a challenge then. If we are there, we should be able to build a lot, like going back to Feynman's quote.
I might interject for just a second. I think I totally agree with Celeste, and I think that she, the slide that she had at the end showing how nature has to choose a developmental program consistent with the material that it's trying to sculpt, I think is like a really powerful insight. One thing that I do think remains a challenge and is really blocking us from doing a lot of what we would want to do in vitro at least is that sort of as Thomas, I think, beautifully articulated, development always operates on a specific set of initial conditions, and those are set up in a very careful and sort of stereotyped way during development. And so a lot of energy is invested into getting the initial conditions right before the next step of development begins to take place. And although we have tools for doing this in a relatively simple manner in vitro, like we're really good at making spheres, right? Making more complex closed topologies from sheets are something that we don't know how to do very well unless we're putting cells on relatively artificial substrates. And so those closed topologies are fundamental to the development of essentially every organism. And it's clearly hardwired into the way that the sculpting of these materials proceeds during development. And so, how do we do that? What are the tools that we should be exploring to do that? I think is a very important question for us to explore. Is it going to be 3D printing? Is it going to be microscale engineering? Is it going to be synthetic development? Like we're going to put organizers into tissues and let them get to a structure and then like let it go from there? I don't know the answer to that, but I think it's something we need to figure out.
Thomas?
Right. So I was about to actually develop this perspective. What's really striking to me when I look at my colleagues who study, organoids or gastruloids, as they are called now, is they are facing the lack of specification of boundary conditions, initial conditions. And so you might say, well, actually, they will discover those boundary conditions and how important they are. And certainly they have improved their protocols along these directions. But in the end, when they manage to actually canalize the outputs to actually drive the systems to where they want, in the end, I think what's also interesting is that in development, it's not just that you inherit boundary conditions that canalize a trajectory. It's also that the system itself actually updates its boundary conditions in ways that we don't understand. It's a highly nonlinear process if indeed not just you inherit them, but you modify them. Yeah. I hesitated to actually talk about another story where actually you have similar setting, but actually the boundary condition is also an ECM gel. It's actually the tissue that Jitu told us about, the wing disc. The ECM around it actually is growing as a material and is growing in very interesting ways. It has anisotropy of growth. We don't understand those, but I imagine that more generally, having control over the trajectories to give an end result will require an understanding of not just how you start with initial conditions, but how you update them, how the system can actually control its own boundary conditions. So it's not just that you add them as an experimentalist, but the system in development actually does do it. So my question for those like Celeste, Amy, and Yusef who actually work on this bottom-up system in vitro is, do you have an understanding when you put matrix whether actually the cells themselves actually secrete their own ECM, modify what you added in the beginning? Because I think an understanding of that will be necessary to actually have a better understanding of why systems go in a given pathway or not.
Yeah, well, I think one thing what you said sparked for me was just to mention that in the rings, we don't define the width, but it is determined by things like the cell density. So if we start with more cells, we get a wider ring. And so the cells have their own internal sensing mechanism to establish the geometry that suits the spacing they're searching for. And again, that width has a really high effect, a strong effect on the pattern itself. So yeah, the way that the cells take a geometry they're started with and then evolve it to where they need to be has a really strong bearing on the pattern that forms. But we don't understand how they're sensing that or how they're establishing that.
Celeste?
Yeah, sorry, I'm listening to Amy's response. I mean, certainly to get to the very specific question that Tomas just asked, when you stick cells in Matrigel, and I'm just going to use a general cell here, of course they remodel their surroundings, and so absolutely one needs to have some understanding of the dynamics by which the cells are modifying the environment in which they're located. But I think one point that I would like to raise is that development is not just morphogenesis. So to get to Ron's original question and sort of the original positing for this discussion by Zev and Amy, mechanism. The word mechanism, there's lots of folks have lots of different definitions for what mechanism means, and in order to address that, you really have to know what you're talking about. So or come to some agreement on what you're talking about. So mechanism for what, right? So development and tissue structure and morphogenesis is just one part of that. The other part is that these cells are actively differentiating and changing their fate as these tissues are constructing themselves, and as we try to construct tissues in culture, and that by itself, those processes by themselves, I think we need a richer toolbox than we currently have. So I don't know if that addresses your question, Thomas, but I think that's one point that we haven't really discussed so far is this connection, this needed connection between morphogenesis and form and tissue morphology, as well as the dynamic changes in differentiated fate of the cells that are occupying that tissue.
Actually, yeah, I wanted to comment on something that might relate to extracting like general principles of tissue organization. I really enjoyed, Celeste, in your talk, the fact of looking at evolutionary distinct models. And I wonder how much we could think about physiology in different systems across evolution to then correlate it with the different shapes that they have in those systems, like in that case, the respiratory system, but the cardiovascular system is completely different depending on how the physiology is adapted. So something I think we haven't touched upon, but.
Okay, how about you?
Yeah, no, I think Celeste, really articulated the key missing piece. While one may have the toolbox for generating structure and form, the underlying, cellular processes and machinery is, as well as the constraints on what the cells and the possibilities that the cells can offer is I feel a big black box right now in this field. So, connecting that to the, possibilities of looking at at the creation of structure, form, and differentiation, to me is the key, big, open question. And effective models like the ones that Zev also talked about, and being able to link those to cellular activities will provide, I feel, a deep mechanistic understanding. That was just a comment I had.
I actually follow what Ron just brought up, like building something. I kind of wondering, like people thought the aim of building something, whether we want to build an organ like an animal or human organ, or we want to build something that actually function like or can achieve certain level of function as a human organ. If it's the later one, then whether we need to make it as close to human, or we actually prioritize on which part of the organ has the most important function and try to recapitulate that part. And yeah, just curious about people's thought. Yeah, I kind of like leaning towards the second one.
I think that's a really important point, right? Because we know, for example, that kidney function can be considerably degraded and you can still walk around and function totally normally. Mm-hmm. It's just sort of in the extreme cases that you need 100% function. And I think we can extrapolate from that the kidney probably isn't perfect from a design perspective. You could probably do a whole lot better. It's just that maybe for some reason, with the tools available to it, evolution couldn't do much better than where we're at right now, or there just isn't enough selective pressure to get us any better. I think the sort of flip side of your point is that just thinking from the perspective of form, I think that there's many ways of getting to the same form. And I do think it's an important question to ask, to what extent is the path that you take important for the final function? We tend to look at, I'm going to call myself a tissue engineer for a moment. We tend to look at the scale of tissues and cells to see whether things look the same. But there's a lot that's hidden from us in terms of the alignment and the composition of the ECM, the diffusible factors, the metabolic states. Sometimes it's hard to see whether those are the same in our engineered tissues.
Yeah, I just want to comment on that. I mean, like my dream is basically like exactly the latter, right? So like focus on the function, build some form, short circuit, whatever the complex, normal developmental biology might be doing, but like try to grasp the key points and like achieve that part of the function. So I guess, well, maybe Meng was about to ask that question, but I will steal that question. Is what do you think is missing, so that we can try to like connect the metabolism part, the energy, bioenergy to all this signaling and mechanics, right? Because the mechanics and the biochemistry somehow is kind of like exhibiting in the outside, but inside the cells, a lot of process will still have to consume this energy. And do this just like naturally fall together? Do the cells just like are smart enough to be like self-tuning to say like, now we need more ATP, we just do more ATP, or do they have like some other, I don't know, like ways you have to tell them?
Yeah, I have a feeling the next time we all get together, we're going to know a good bit more about that. It feels like something in the ether exists that is inspiring many to start to ask those questions. So I think it's an important frontier that I imagine there'll be some progress on in the coming months and years.
That was still part of my question, so I still have another part. So for me, I'm always intrigued how organ size correlated, with the body size. So it seems like the organ know when it should stop. The morphogenesis part, and now is the final point. I reach to the stop, and also I think it's even more intriguing to me. Sometimes when I think about liver regeneration, whenever you cut it, it grows to the size. It's not bigger or smaller. Mm-hmm. So how the organ know? So what? Also, like when we look at size of different organs, in a way they are proportional. It's not like we have a gigantic lung. So how the tissue know, to coordinate this? So I think that's like the part that Xiaohe still is like whether energy flow in the body is a factor to tell them.
Okay. Sorry, go ahead, Celeste.
Oh, I was going to say, I think that's a beautiful question. It's a wonderful question. I think for most systems, we have no idea what causes it to stop, right? And this is where I get really excited with looking at non-canonical, non-standard model systems to kind of, address this question. So we recently started in collaboration with Ricardo Mallarina's lab here at Princeton to look at a model marsupial where there is a disconnect between the development and the size, the development of internal organs and the size of the developing embryo, but because of the lifestyle by which marsupials are born and then have to continue developing as joeys. So I think, some of these questions with respect to size and stopping are going to be really, really challenging to investigate if we focus only on a single species or only on a single system. But we'll get more insight by looking at the differences and the diversity that's present amongst the species that are available to us.
Yeah. So just to build on some of the points that were already raised, we continually are talking about ECM and extracellular matrix, and I guess this is a more practical. What do we do next on this topic? Because it seems to me that, as a developmental neurobiologist, a lot of this is about timing and context, right? And you could basically pick any time point through development, past morphogenesis, all the way through to refinement. And the local signals at the level of ECM and secreted signals from other cell types, as well as vascular signals, are going to change. So I guess on the ECM front, since this seems like a very conserved, based on the data we just heard from Zev and others, what could be done there to systematically study and test ECM in a way that is attractable now? I mean, every species, every time point, every model is going to have slightly differences there, but it sounds like there's something that could be done beyond Matrigel where you can start to systematically look at how conserved these different ECM complexes are and proteins, what is unique to different stages. I just wondered, this is not my field, I wonder what everyone thought about that, because it's come up multiple times over the last couple of days, and it seems pretty important.
Yeah, I mean, I could throw something out there. I feel that, yes, it's super important, right? And it's certainly, and it's been recognized as being important for 30, 40 years, right? I think there's a couple critical roadblocks. I think for one, until recently, sort of gain of function genetic perturbation of the ECM has been incredibly challenging because these are humongous molecules, right? So you can knock things out, but there's a lot of compensation. But like, how do you do the gain of function experiment? I think another important conceptual roadblock is, as I think Celeste nicely put, and Thomas was insinuating with this question, that the cell ECM interface is a living interface. And so you can put cells in an ECM, and for a short period of time, they might get sent off in the wrong direction. But ultimately, they're going to remodel that interface because they're trying to reinforce. This is a concept Mina Bissell put out there now 40 years ago. They're trying to reinforce their external environment. It's some reflection of their internal state. So like thinking about that makes the sort of microenvironmental perturbations that we might imagine challenging. So where to go? I think one, we don't know. We know next to nothing about the dynamics of the ECM. We know next to nothing about the properties of cells and ECM as a composite active material. We know next to nothing about like all the invisible molecules in the extracellular space that are bound up by the ECM because it's hard to see them by fluorescence microscopy, which is our primary tool.
Is this where proteomics, some of the approaches we heard earlier, can start to be applied? Where in the same way we're sort of unbiased sequencing, brains and transcriptomes, to start to apply that to ECM and use models and in different model organisms to get at least the parts, as Celeste said, to build on to get more of the high resolution imaging tools to fire to?
I would certainly say that we need the parts list. And we also need to know something about the dynamics because there's almost nothing known about that. I'm curious what Amy and Celeste think about this.
We're giving it a shot with the proteomics. I think it's a good idea. And I agree that we're at a point where a parts list would be useful, even if or beyond that in other aspects of signaling and sort of the physics of cells. I guess I want to be mindful of our time, so maybe it looked like there was maybe a couple more questions, and then we could continue over lunch.
So I wanted to push the idea that you said there are many ways to get to the same form. So, but how about function in intermediate forms, right? Because in development, you have to remember that the organism is still trying to survive and adapt to the environment. So all these intermediate forms in development still have a function. Same with repair, for example. So it's not just repairing the original function, the damaged tissue still has to function. So I don't know if we're doing more on investigating functions in these intermediate forms because they constrain part of the boundary conditions.
Yeah, I think that's a very interesting question.
Yeah, and there's certainly examples in development, for example, where a form will develop and it's a marker, it's a foundation for another step in development, but it ultimately goes away, right? So its function is purely defined in the context of setting up road landmarks for development. Whether the same can be said for heart pumping and intestinal function, that's a really interesting question.
I think the lung is a pretty interesting form for that, Celeste. I don't know. It's probably something you've thought about.
Well, the lung is interesting because it's not being used as it's developing, right? Like there really is no actual function of the embryonic lung, unlike the heart, which is contracting as it's forming, and all of the other internal organs which are starting to do their ultimate mature function. And the lung I find incredibly fascinating because if we just take a look at the mammalian system, it's not the most efficient of gas exchangers, but the final architecture is actually pretty highly optimized to reduce the energy necessary in order for air to flow through all of those tubes. And somehow it's optimized when it's not being used for its function. So, I don't know how to address the question of a form being used in the context of an organ which isn't actually useful to the embryo in any sort of way that we know. But I think we're going to probably find lots of different answers to that specific question, depending on the system that you look at.
Okay, we're going to do two more questions. So Ruslan and then Thomas, and then lunch.
If your question is relevant, you go ahead first. Yeah. Well, I just wanted to get back to Ron's question, because as an outsider to development biology, I also feel like we know a lot of really beautiful details about individual organ developments, but I really craving to see what are the common themes and principles are, and I cannot find it anywhere in any textbooks, reviews, and so on. Maybe I haven't read the right ones, but I tried to find something that will put it into a perspective where it's described from first principles. These are the common themes. These are variations on themes. That's how you get whatever, lung versus kidney and so forth. And the great details that we know from the work of people in the field filled out a lot of very critical genes and pathways and how they regulate each other. But are we at the point where we can use symbolic representation, or you remove all these morphogen names, put symbols, and say, well, these are the types of connections. If you have them in place, you get invagination, you get lumen, you get branching, and so forth. And I don't know if that knowledge exists and not expressed anywhere, but I feel, again, as a total outsider and completely ignorant about all of it, but I feel like that there hasn't been a description from first principles defining the common themes, what are the number of themes, perhaps there are more than one, and how we get different specific programs as variations on those themes. And that's just my very subjective, ignorant opinion.
So is that a call to arms to put together a new textbook?
Well, there are no textbooks, but I would actually argue that for someone who has been working on morphogenesis for 25 years, including my PhD, working on morphogens and things like that, I think what has been a revolution from my point of view in the last 10 years is really that through the interface in collaboration between physicists and developmental biologists, there is now a very rich conceptual framework to think about any developmental morphogenic process from first principles. We've probably failed dramatically to communicate that.
Yeah, where do we find it?
But no, I'm most happy to discuss with you, but I think there are a number of reviews that already have communicated that. I think we can do that. I mean, I'm definitely interested in principles, and we are extracted those principles from the comparisons of many systems, precisely. So but that was not my point.
But it seems like there is abundance of examples, very detailed examples, but okay, maybe you can tell me.
I wanted to say two things. One is I want to amplify what was said before about size control. I think it is one of the biggest unanswered problem in the whole of, I would even dare saying biology, because I think it's a huge problem. I don't know whether anyone at 4DCP wants to tackle that at some point. I think, yeah. But I wanted to bring another sort of technical problem. I mean, it's really embarrassing for those who are interested in morphogenesis that whenever we want to even study shape, like think of a 2D surface embedded in 3D, and you want to see how it evolves, okay? We do not have good mathematical computational tools to actually characterize those shapes in ways that you can compare a shape in one experiment day on Monday and another experiment done on Wednesday from a lab in the US and a lab in France, because differential geometry is not a simple field to embrace when you're a developmental biologist. Even physicists are not all well trained for that. How do we build like average structures? How do we map these complex geometries onto activities? I think this is an area where actually data-driven approaches will lead the way through the collaboration between computational scientists, computer graphics, people like what Gaudens Danuser is doing, for instance, theoretical physicists and biologists. I think the field is in demand of such approaches. I don't know what Celeste, Amy, Zev, and all the others think, but I think this is crucially missing for the field to actually study shapes emergence.
Yeah, it falls into almost a category of phenomenology, which Zev and I were talking about has been almost a dirty word for a while, but I think sort of reappreciation of the importance of just grasping form and shape, I think, is an important thing.
But the reason why I bring it is maybe at Janelia, given all that is done at the organelle level, at the cellular level, there ought to be, maybe some of the things that have been learned as tools could be, used fruitfully to actually address, same problems at the larger scale.
Yeah. Unfortunately, we have to sort of end this amazing discussion. But I think it's been basically this discussion, I think, really has highlighted what we see as the need for a 4DCP focus in this kind of broad way. Basically, we've heard, brain-body, impact on, physiology. We've heard about mechanical impact on physiology as well as morphology. And we've heard a bit about metabolism. We've also heard about tools, tools that allow you to probe particular, biochemical states, probes that allow you to look at the morphological states in ways that we just could not have imagined 5 years ago even. So I think that, this symposium has reinforced my just incredible enthusiasm for this field. I know Ron feels the same way. We are just so excited to be able to be a place where we can do this type of work with new technology all around us. But I also want to highlight something that I think Ron mentioned at the beginning of this session, is that we also want to make the 4DCP effort at Janelia something that's worldwide, something that all of you can participate in. So we're hoping to be able to carry out activities, ongoing activities that can bring in other people to focus on this really incredible area of 4DCP that I think is just ripe right now for investigation. So I just want to sort of highlight some things, just in conclusion, just highlight things that moving forward, how we might try to accomplish this. One way is to have another meeting like this. And so, in fact, Janine has done a fantastic job, not only for this meeting, but she's already starting planning for another meeting, which we're planning to have a year from now, October 15th to 18th. So put that on your calendar, and hopefully we can get everybody back again to really talk about where we're moving in this field. Hopefully we'll have refined some of the visions, some of the big questions and issues. I think it's been just spectacular the last 2 days to be able to see the breadth of this field. I also want to invite everybody who has a cell biology focus to join